Okay, well, as I say on this slide here, immune therapy really has dominated here at ASH this year, and in each of the three areas that I list, the CAR T therapies, the bispecific T-cell engagers, and then the monoclonal antibody drug conjugate, the GSK belamaf, a lot of interest in the updated data there. Mostly because all three categories are moving forward, and we're expecting that they will have some degree of approval in the near term.
Yeah.
So first of all, for the CAR T, obviously the CAR T therapy, we've been thinking as a possible game changer for the last couple of years or so. This harvesting of the patient's own T cells and the genetic engineering. The concerns have been obviously that it takes time to do that, so that you have to wait for 2-3 weeks, and then you know, the concern about the toxicity, particularly cytokine release syndrome. So there's been a lot of promise, but also some concerns. However, here at ASH, there was I think probably a big sigh of relief, especially from the Janssen company, that the product from the Legend Chinese company had a very successful trial here in the U.S. With a very favorable toxicity profile. You see here that there was one patient who did have some dire complications, but otherwise the profile of the toxicity really quite acceptable. And then, of course, the deep early responses really excellent, and so I would have to say tremendous enthusiasm about that, which was actually reflected by the fact that they did in fact this past week receive a breakthrough status from the FDA. So I think that everyone is kind of feeling this impact of this promising therapy. Now, in addition to that, there were a whole range of CAR T protocols, and it's hard to pick one, but I just thought I'd point out something which is a little bit of a trend, so that the Cartitude came from China, but at this meeting there was also a second one that comes from China that was presented, which has a safety switch in it. And then there's another one coming along, which is a humanized version. So we're seeing this trend where some of these new innovations in the CAR T space are actually starting off in China, and with some validation that we're seeing. And so it was interesting to see that this Ichran product showed very excellent results in 3 different hospitals with excellent responses and with acceptable toxicity. And it was really encouraging to see similar PFS and survival data. And also interesting to me is that they also are starting to look carefully at why is it that some patients are non-responsive or relapsing, and they made this observation, which has not been made before, that it could be linked in with the mTOR pathway. So some very interesting things happening in that arena. But I would say that maybe even more excitement, because people have been hoping to see what would happen with the B-cell engagers, T-cell engagers. And so this is the product from Celgene, and early data showing, again, using this, which is similar to a BiTE. BiTEs come from Amgen. This is basically like a BiTE, but coming from Celgene. And you can see strikingly here that there was a strong dose effect. At the lower doses, not too much benefit, but they have achieved a very effective dose with acceptable toxicity, and so clearly strong interest in that. And so perhaps a different type of excitement about the monoclonal antibody drug conjugate, the GSK product, because it might possibly be the closest to coming to commercial availability. And so obviously 2 or 3 things to point out here. Obviously there is a direct monoclonal antibody against the same BCMA target, But the interesting point here is that it's not so dependent on the BCMA expression, because if you have binding, then with the introduction of the drug into the cell, you have that added cell kill potential, maybe even at lower BCMA expression levels. So a number of areas of interest there. But again, here we have a strong response profile, maybe slightly lower than the CAR-T and the bispecifics, but very impressive responses and durations of response. The unusual thing about the GSK product is the corneal toxicity, which is definitely going to have to be carefully managed, but it does seem to be manageable and reversible, and so it seems that this will be an issue that we need to be aware of, but something that can be handled. And so, what do we think about all of this? We've got 3 groups of agents, all clearly very active in the relapse population. And so how will we look to introduce those for our patients, to sequence them potentially? And in this case, do we see them having a best use maybe earlier in the disease for consolidation or an earlier relapse situation? And so, Mary V, maybe you want to start first. What do you think about this?
Well, definitely I think that all new immunotherapy strategies you have shown, especially CAR-T and the T-cell engagers, are encouraging, and I would say that even in the future I can envision that they can be complementary.
Yes.
Why? Because the results from CAR-T are spectacular, but it's true that in myeloma, for me the problem is the duration of the response.
Yes.
And the duration of the response is not so long as expected.
Right.
Maybe CAR-T is excellent in order to achieve rapid and deep responses, but we need something to maintain the response.
Right. I agree entirely. If we look at the PFS curves, I showed you a couple there, and there is a continued drop in those curves.
So why not to incorporate the T-cell engagers as part of maintenance after CAR-T?
Absolutely, or even the GSK product.
Or even the GSK. This is a complementary strategy. The results you have shown about the T-cell engager are really comparable to that reported with CAR-Ts.
Yes.
Also rapid and deep responses. We need a longer follow-up in order to see what is the durability, but if the results would be similar, these T-cell engagers would be ideally for patients non-eligible for CAR-Ts.
Right.
Very elderly patients with some comorbidities, poor performance status, or frail by definition. These patients would be candidates to the T-cell engagers or maybe the belamaf madofotin.
Correct.
With the belamaf madofotin, we have, as you said, we have to take careful about the ocular toxicity. And also we have to take care about the thrombocytopenia.
Yes.
Because patients develop thrombocytopenia, and usually it takes some weeks, even some months in order to recover it.
Okay.
But definitely I think that the results they showed are promising. I think that all these drugs, all these modalities will be moved earlier on, of course. Mm-hmm. And we need, of course, more data and more clinical trials in order to know what is the appropriate therapy and at which time, and how are we going to incorporate all these new strategies in the landscape we have just been discussing about quadruple combinations based on VRD or KRD plus data.
Right.
How are we going to combine?
Right. Absolutely.
The good thing is we have more options, right? And this is what I think is the most important.
That's the good news and the bad news.
Yeah, absolutely. We may not know the right order and the sequence, but I think the more the better.
Yeah.
The 2 things I'd add to this, Brian and Marie V, one would be, I don't know if anyone really feels that the CAR-T products we're using right now are going to be the final ones we're using.
Yeah,
so we're seeing
this is a very important point.
As I like to say, the next generation cars, right?
We're getting starting point.
Exactly. We're getting better horsepower, and we're adding a bigger moonroof, and we're having better features to the car, and I think that's helpful.
One philosophic part to that is, if we're looking for a better car, do we want one where it's used in a decisive way, or one where we're looking for a more prolonged exposure? Are you with me? If it's humanized and might be given. My bias is that we should maybe be looking for that decisive impact.
Agreed.
Completely
agree.
As a new CAR.
Almost like a potentially with the chance to replace transplant at some point with time, but where it is very intensive. But if these new CARs, if you will, can persist longer, and I think of this as the hybrids, right? They can go longer. I know the analogy could be stretched here, but one of the reasons why we think, at least, that the current CAR T-cell strategies aren't lasting in myeloma patients is their T-cells are exhausted. Their T-cells don't have the longevity.
Yeah.
You know, we're taking T-cells that are kind of at the end of their life cycle and expecting them to
so
to
using
go back
cells harvested earlier in the disease, right?
Correct. Or if we have other ways to you know, give those T-cells a bit of the fountain of youth to stay on would be good.
Yeah.
The second point is, I think another very important reason why we want these different options, especially with, you know, belantamab mafodotin and the bispecific agents, is that even if it is that decisive a strategy, CAR T-cell therapy is always going to be difficult to have greater access.
Right,
I mean,
absolutely.
right now,
Intrinsically,
we
always,
think, exactly, maybe 30 to 50% of people who could get a simple autologous stem cell transplant in this country don't get it, because they may live too far away from a transplant center, or there's travel issues or insurance issues, whatever else. You know, CAR T-cell therapy is not exactly like an autologous stem cell transplant, but a lot of similarities. CAR T-cells,
and so
it is a bit of a boutique approach. So
having a fantastic bite could be
correct to sort of take it off the shelf,
Absolutely.
right? So a community oncologist can, you know, literally infuse this drug over an hour, as opposed to going through the weeks of collecting the T cells, manufacturing, waiting for them to come back, reinfusing them. That may be, especially as you mentioned, in a more frail population or one in whom they may not be able to have that intensity. And obviously the time will tell us if there is really a superiority of one over the other. All we can say is we've not seen this kind of deep response rates this late in the disease course with anything else.
Yeah,
so they're all three of them are very impressive.
Right. And I think that the other thing is that we need much more information about, for those patients who are progressing, you know, why is that? What are the mechanisms of resistance? Is it a mechanism that could be overcome by giving more cells, or is it something that where we need to give a different type of therapy to achieve a better outcome, you know?
Agreed.
Or perhaps I don't think we know too much about that yet.
Yeah, a maintenance approach after of some sort. Yeah.
This is something that we don't know, but definitely, well, during this ASH, we had the opportunity to see an abstract using gamma secretase inhibitor in order to upregulate the BCMA expression on the plasma cell that can be excellent complement to the CAR T when the BCMA expression is lower. Yeah, exactly. And I think that definitely we are going to have the possibility of to manipulate, to generate new generation of CAR Ts and to complement with all drugs that we have. Maybe even it would be possible to incorporate again in myeloma the checkpoint inhibitors as complementary strategy. We see that one of the mechanisms of escape can be potentially overexpression of PD-1 and PD-L1.
Yeah, it would be good to be able to bring those back into the mix in a safe way.
Exactly.
And I know this is something that's sensitive for you.
Yeah.